And the more time it goes, the more convinced I am becoming that research scientists, in one way or another, are neurodivergent. Let me explain. For a person to dedicate most of their lives to one particular topic and become so obsessed with that topic in order to make groundbreaking discoveries, you do need to have a different way of thinking and different way of being than your, normal people. And in comparison, most of the earth is made up of, neurotypical people. The smaller percentage of those folks who are research scientists, if you look at their lives, it's really fascinating because they tend to forget everything else, or most of the things that happens in life, and they just keep being very focused on that one particular topic. And that's also one of the main reasons that they end up making groundbreaking discoveries. And even if it's not groundbreaking, they still make discoveries. And so you don't need to be a Nobel Prize winner to make those discoveries. A lot of scientists do that on a daily basis, but they're still, in my opinion, the way I see it, must have some type of a non-neurotypical way of looking at things. And today, I want to share with you a paper that came out regarding the electrodynamic theory of life. And there were two scientists that work on it back in 1930s, Burr and Northrop. And I'll add the link if you want to read the whole paper on your own as well. Harold Saxton Burr was a biologist working throughout the 1930s and 1950s on an important set of problems related to biological organization and the origin of complex living forms. He was a profound thinker, suggesting a complementary focus on field concepts, in addition to the emphasis on particle models and integrating concepts from physics and philosophy in his work. He developed innovations in electrical physiological techniques and used them to perform a wide experimental survey of bioelectricity in normal and pathological growth. And Burr ended up writing a classic paper in collaboration with the philosopher Northrop called The Electro-Dynamic Theory of Life. Decades later, it is now clear that Burr was a persistent and visionary thinker. His main hypothesis that bioelectric gradients serve as pre-patterns guiding morphogenesis has been confirmed using modern molecular physiology, as well as his idea about the place of cancer and the nervous system in the question of biological organization. With limited technology but deep insights, he derived insights that anticipated many modern discoveries. Even more importantly, Burr's view of bioelectricity as a convenient entry point for rigorous investigation of the broader question of self-organization properties of life highlights a frontier of inquiry that awaits today's researchers. Burr and Northrop, the electrical dynamic theory of life. Throughout history, there have been thinkers who displayed a remarkable degree of persistence, deriving a vision of the world that is ultimately borne out by scientific advances long after they are gone. from very limited information and tools available at the time. Harold Saxon Burr was one of those scientists, a tireless researcher. He worked at the Yale University School of Medicine and published 5 decades worth of primary papers and opinions and analysis pieces. He is a founding figure in developmental bioelectricity, an interdisciplinary field that now spans areas including the origin of multicellularity, the relationship between genome and anatomy, regenerative medicine, bioengineering, soft body robotics, and basal cognition. Burr was fascinated by bioelectricity, like me, which is the voltages and the electrical field produced naturally within living tissues. These bioelectrical signals derive from the activity of ion channel and pump proteins in cell membranes and spread throughout the cell networks via electrical synapses known as gap junctions. But Burr didn't know any of this at the time that he was active. The molecular source of bioelectrical gradients was not known at that time, and the relationship between the heredity and the cell function was just beginning to be worked out. He produced the first millivolt meter usable for investigation of bioelectricity and used it to explore a wide sample of biospheres. For example, as early as 1955, he had already seen the promise of unusual model systems such as slime molds, which have recently become popular contexts for understanding the decision-making capacities for cells and the role of biophysical forces therein. Extremely fascinating. While interest in recording the bioelectrical correlates of morphogenesis was already apparent by 1903, Burr's work was the only data set big enough to permit generalization beyond one organism. Burr quantitatively recorded and analyzed the bioelectric properties of embryos, adult organisms, tumors, and trees of a wide range of species, including human beings. As a kind of informal meta-analysis of these extensive measurements, he thought deeply about the meaning of the universal occurrence of spatio-temporal patterns of bioelectrical states in diverse tissues, like me, and the fact that changes in these states correlate with important morphogenetic and patterning events. Techniques for testing the functional role of these gradients had begun to be available at the time. Indeed, people, including Morgan, were already applying electrical field to developing and regenerating systems as early as 1904. However, Burr was well aware of the difficulty in making predictable changes to indigenous bioelectrical properties with applied electrode approaches. He analyzed the measurement data from both neural and non-neural tissues and proposed that the distributed pattern of electrical potential served as a kind of a scaffold, which is a pre-pattern that guides cell behavior and thus controls growth and form. The Electro-Dynamic Theory of Life paper begins with a philosophical stage setting, arguing for the greater emphasis on organization in addition to entities, a concern that is increasingly relevant in today's debates in system biology and the exhausting profiling of data. Burr discusses the utility of field concepts as a complement to what was already in his day apparent as the overwhelming direction of biological research, chemistry. These models focused on purely local interactions at the time. He saw earlier than most of other researchers that an important aspect of understanding biological systems was a rigorous appreciation of the non-local nature of control in morphogenesis and physiology. Burr weaves in concepts from physics, discussing the bidirectional feedback between field and particle. This paper is focused on bioelectric field as a specific example, but this is a much bigger issue currently playing out in the state-of-the-art discussions about agent-based models and downward causation from global virtual governors and global distributed dynamics that emerge from relatively simple local rules governing proteins, subcellular structures, cells, and tissues. Burr's work in general received limited attention. As molecular biology took off, most researchers gravitated towards emphasis on the more easily studied biochemical and genetic mechanisms, which, unlike bioelectricity, could be studied in fixed, chemically fractioned preparations. This, of course, was in line with precisely what Burr was warned about, an overly exclusive focus on those aspects of cellular structure and function that could be studied in isolation at the expense of holistic aspect studying. Even prominent thinkers who were making similar points at the time did not publicly support Burr's insight that bioelectricity represented a set of mechanisms that would in time become an immensely tractable entry point into supporting the organistics philosophical positions. And decades saw an explosion of work on ion currents and electrical fields. It was not until 1970s that indigenous voltage was shown to be critical for single cell bicone. And not until 2000 that the first tools were created to examine the role of voltage in a multicellular context, using fluorescence molecules that function as in vivo voltmeters. In the last 20 years, a variety of Burr's specific predictions have come true. Spatio-temporal patterns of bioelectrical gradients are indeed pre-patterns that, along with chemical gradients and biomechanical forces, specify large-scale anatomy of the body. This has been shown in the context of eye development, brain shape, skin coloration, left-right axial patterning, and anterior-posterior axial patterning. Pre-patterns of bioelectrical properties that predict large-scale patterning in organisms have been studied and functionality implicated using molecular tools. Moreover, it is now clear that the bioelectrical signaling layer is an important aspect of physiological software that enables genome specified hardware to exhibit the remarkable plasticity observed during regeneration and remodeling. Confirmed have been a variety of Burr's ideas about the relationship between the nervous system and the non-neural bioelectricity. Neural structures are indeed exquisitely sensitive to bioelectrical cues provided by the neighboring tissues, as he predicted, seen by the modulation of host cell electric potentials to control outgrowth of transplanted neurons, and by the patterning of the brain by pre-existing bioelectrical patterns. Similarly, Burr's idea that the nervous system carries a significant portion of instructive bioelectrical signaling has recently been confirmed in the discovery of the brain's directing of tissue morphogenesis in development via ionic signaling, and the role of the central nervous system is directing the regeneration polarity. Burr even partially anticipated the computational neuroscience techniques being applied to non-neural bioelectricity by inquiring about the design principle of the brain and the information processing functions. Consistently with Burr's ideas of the evolutionary process shaping brain and somatic tissues, it is now increasingly beginning to be appreciated how neural computation evolved by speed optimizing information processing tasks that cells were already doing long before nervous system evolved. Burr also correctly identified cancer as the flip side of multicellular organization, equally dependent on bioelectrical cues. Moreover, he found that the disturbances of normal bioelectrical patterns can be detected as long range, presaging by more than 70 years of discovery that non-local imposition of bioelectrical states by different techniques can normalize cancer. It is clear that the evolution discovered the unique properties of bioelectrical circuits, implementing the communication needed to bind component individual cells towards a common organ-level morphogenetic purpose as far back as bacterial biofilms. Had a remarkable ability to foresee advances in this field, and his work was no doubt an inspiration to the major figures in this area who advanced the science in the 20th century. If he were alive today, Burr would have thrived in the context of the revolution of what we're seeing today, such as in the bioelectrical field, that allow modulating tissue bioelectrical patterns at will. Given his ability to see beyond the current mainstream and the willingness to embrace and extend the latest technology, we can only try to imagine the advances he would have made at the interface of molecular biology and developmental physiology. Due to his interest in philosophical issues of explanation in biology and the use of mathematical global field models as a complement to particles and forces, he would have been right at home with today's questions about the relationship between DNA, biochemistry, and dynamic form. Interestingly, despite all of the progress since his work, Burr's emphasis on field models of bioelectrical control have not yet been developed. Moving beyond local models of electrophysiology in development and regeneration represents an important area of future work. Burr was an important thinker and pioneer working at the very edge of contemporary technology to produce new data and extrapolating to ideas that wouldn't be properly appreciated for decades thereafter. Equally important as the mechanistic insights into bioelectricity was a thread running through all of Burr's work. And so if you notice this is the type of personality that I was talking about, a researcher who's dedicated most of his life, and many other researchers have done the same. And you literally cannot come to that level of focus if you are giving your focus to everything else in life. And I'm not saying that that's a good or bad thing. I'm just being an observant here. And I think that's the reason that I am beginning to believe more and more that a lot of these researchers have some type of a neurodivergent personalities, because it's very hard to remain focused on one single area, especially when the rest of the world, especially those that you respect, let's say your peers and your other researchers in the field, etc., they don't pay attention to it. You need to be convinced enough and focused enough and have enough confidence to believe in your own intuition and in your own level of seeing the patterns that others don't see to continue. Now, obviously, he was at a very prestigious university, and that's obviously a great sign and a great support system there. Nonetheless, a lot of times, even when you are in those situations, you have a very, very talented pool of people whom you're working with and in parallel. And when they don't recognize your work or don't pay attention to it, and sometimes, most of the times, even ridicule it, it's very hard to continue. And I have a feeling that a lot of these folks are so convinced about their work and so focused that nothing really matters. And I think that's the superpower of being neurodivergent. We see that with a lot of neurodivergent people. The only thing that, in this case, the only challenge of a neurodivergent person is to keep continuing the work and not get frustrated, because sometimes you're not getting there and you're just like extremely frustrated, or to use the tools that you have and just move on and maybe take a nap or go for sleep or something like that, and wake up in the morning and just be refreshed and keep going, because it's very important to keep the focus and not get frustrated and just keep moving. And we sometimes get to the finish line and sometimes we don't. We just never know. And but it really, really, really needs a specific type of personality to stick with a focus that you have and just dedicate it, and maybe take the focus away from everything else that this world values, because your value is different than them. Like, for example, go after money and be a business person and things like that. So that's why sometimes you see researchers who are really dedicated, they really have a humble life. They don't make a lot of money at the universities, for example, or even when they're independent like me. But it's just that they're just happy with the work that they do, so that their value is set differently. And if they really want this very luxurious life, sometimes it's not possible, because that means that you have to switch off what you're really passionate about and what you're focusing on, and go and be a business person instead. There's nothing wrong with that. It's just that it's a way to choose different values and emphasize the different values that you have in your life. And sometimes it's really hard because you want both, but it's like you can't. And one last thing I want to say about this focus is that for those of us who are really focused on this one particular topic, let's say, and we value that, it's not that, sometimes it's not that we want to, it's just really hard even to switch it off. You know, we can go to bed and still thinking about it. That's like all we're thinking about. And that is also the reason sometimes it gets really frustrating, because maybe we don't have the luxury of having the tools that we want, having the labs that we want, having the support that we want, and it just goes very, very slowly. And so that's another reason I wanted to read this type of papers, to kind of remind ourselves that it's okay, and we're all here for different reasons on this planet, for whatever reason it is. And if this is your reason, definitely you should go for it. So that's all I had for today. Hope you like this topic. Feel free to share, subscribe, and like to increase the engagement of this channel. I appreciate it. Thanks. Bye.